I'm going to talk about how good and bad odors are represented in the brain. So I want you to put yourself in a different mindset, think about what smells good and what smells bad for you. And what we're going to talk about is how this is manifested in the brain and how it's expressed in behavior. And I'm going to use Drosophila as the system to look at this. One important thing to understand a lot about what I'm saying, look at the brown sort of bulbous part in the middle of this Drosophila brain. That's the olfactory center. Your center looks about the same. It's composed of these little spherical things. They're all called glomeruli. And each glomerulus takes care of information regarding certain kinds of odors. So that's quite important to know as a background. I gave a certain kind of title. I'm going to start talking about that, but then I'll drift over into something slightly different, I'll tell you. What we're mainly interested in is how smelling works, how does olfaction work overall, and how does it connect to behavior, and how does it evolve. So more or less from receptor to behavior. What you see here is the amazing variety of olfactory structures in insects. So insects smell with their antennae and with their palps. What you see below in the black and white pictures, those are sensilla. Sensilla are the units that insects smell with. So on the antenna you have a lot of hairs. Each hair is innervated by 2, 3, 5 neurons, and they pick up different kinds of neurons. They express different receptors. What you see down to the right in purple, that's the brain, and there you see in green the antennal lobes that are the primary olfactory centers of the brain. We're of course also interested in comparing insects with vertebrates, and the interesting thing is that we look more or less the same. This is probably a result of convergent evolution, but you can draw a lot of general conclusion looking at insect to, for instance, the mouse here. Thanks, Peter Mombers, for the picture. So, working with Drosophila, we turn to Africa because that's where Drosophila comes from, and you see the amazing variety here of what a Drosophila can eat. So they have to be quite wide in their olfactory sense to find what they want to. But there are of course also things that they shouldn't go to. So there are things that smells bad to them. So good or bad, that's what we're going to look at. And if you want to know more about this, what we're dealing with, you find a nice review recently published here in Neuron from last year in November. Okay, so what we're basically trying to understand is how does this fly find this patch of balsamic vinegar that is applied to this little sponge lying down here. So you see them coming, flying, they land. How do they find it with their chemical senses? That's our basic question. So a lot of people have worked with Drosophila for a long time from a genetic, from a physiological point of view, but they didn't care so much regarding behavior. So we started doing behavior. We took 110 odors that were pretty well established as stimuli for the fly, but no one knows really what they do. So we went to these 110 odors, we did a bioassay, and we looked at what is good and what is bad. And on this picture here, blue is good, gray is neutral, and red is bad. And when you start going through these 110 odors after spending about 50,000 flies, then you get a lot of results here. You get some that are really good, some that are medium good, some that are totally neutral, and then you get 6 that are bad. So not so many odors are bad, a lot of odors are good. Then we went into the brain and we entered GCamp into our neurons, and we could look at input, we could look at output by doing optical imaging of calcium dynamics in the brain. We get these kind of patterns, and from this we could draw this conclusion that there are 2 clusters in the antennal lobe, one saying this is bad, one saying this is good. And this is pretty new result. So this is what I planned to talk about from the beginning, but now we're done with that because I have some really new unpublished stuff that I want to go into from here. And this one, here is the conclusions that you find the coding of valence at the output level. So you have a lot of information coming into the primary olfactory centers, but the first place where you can really tell from the patterns that this is good or this is bad, that is after antennal lobe processing. So after processing in the primary brain centers. And this just came out last week in this publication, so you can read more about it there if you want to. So let's go into what I really want to talk about today, and that's something that to the fly smells really bad. So now we've been dealing with odors that are sort of good or sort of bad, but then we found this molecule. And what is this molecule? This molecule is this compound. What is this compound? It's geosmin. geosmin is one of the odors that you are most sensitive to in your nose, and it smells of earth or soil. If you go out after it has rained and you smell the soil, then you get the smell of geosmin in your nose. So it also produced by penicillium fungi, it's produced by streptomyces fungi, and it's produced by blue-green algae. And flies are sometimes exposed to these kind of organisms when they're looking for food. In some earlier experiments from about a year ago, we found in the wind tunnel when we were testing different kinds of compounds and after collaborating with microbiologists, that if you add the geosmin to something that the flies really like wine or balsamico, then you decrease the attraction to 0. So this compound really means something to the fly. So we had something that is really behaviorally relevant to them. So we really know that flies love wine. That's, or they love yeast. They love anything that is fermenting. And that you know yourself, if you come to your kitchen after the day after the party, there might be a few Drosophila in the old glasses. And that's exactly why they really like wine and balsamico. So why are they stopped by geosmin? That's our main question. Why are they stopped? So first thing we had to show, is geosmin really repellent in itself? So we went into T-bar assays like this one, did a lot of them, and we could show that it's really amazingly repellent. The most repellent compound ever found for Drosophila before is benzaldehyde, and that's the purple line here. You see that it's going down. The one black one going up is vinegar. But then you look at the green one, and you have to go like 1,000 times to the left to find this kind of repulsion. Then when you continue this comparison, you see that geosmin is a lot less volatile than benzaldehyde. It's actually 1,000 times less volatile. So not only are they a lot more sensitive to geosmin than to benzaldehyde, but they're again 1,000 times. So they're about 1 million times more sensitive to geosmin than any kind of compound ever found before being repellent to Drosophila. So they are really remarkably sensitive to geosmin. So how is this compound then detected? Because there has to be something specific with this, right? So the first thing we had to ask is, where is it detected? Here is the fly head. In the front you see the antennae, that's the main olfactory organs. Lower down you see the palps, that's the secondary olfactory organs. So flies have 2 noses, you could say. So we did electrophysiology on these, and we could show that it's antennal dependent. They smell it with the antenna, and they smell it with what we call olfactory receptors. I won't go into the details here, but we could show that traditional olfactory receptors pick up this kind of odors. So the question is next, which of these receptors? The fly expresses about 50 receptors on their antenna, so we need to find out which of these are detecting geosmin. And we went into chemometrics because we know a lot of the ligands for Drosophila, contrary to, for instance, vertebrate, mouse, or fish, where we don't know much about the ligands. In Drosophila, we know quite many. So we went in and did chemometrics of all the ones that we know and saw if we could fit geosmin into this pattern of active compounds. There is a lot of programs that you can run on the web, one called Dragon, which takes chemicals, runs the descriptors and groups them according to PCAs or random forest methods. And we got this pattern. If you see here within the gray triangle, that's where all the compounds that has ever been found being ligands for Drosophila neurons, they are grouped within this gray triangle. But then if you look below, there is sort of one black dot, and that's geosmin. So geosmin is unlike anything ever found that Drosophila smells before. It's a totally unique compound. It's also a strange chemical overall. So it's distinct from anything found before, and probably then it's picked up by an OR, by a receptor that we don't know yet what it responds to. So how do we identify such a receptor that we don't know? Well, then we went back and to rely on the imaging methods. So we expressed the GCAMP, calcium imaging, because we know more or less what every little ball in the brain, every glomerulus correspond to on the antenna, because each glomerulus corresponds to one receptor. So we went sort of reversed. We went to the brain to understand what happened in the nose. So after this imaging, we could see that geosmin really triggers one specific glomerulus only. One little ball lights up in the brain when you stimulate with this kind of compound, and it's DA2. DA2 is a glomerulus that is innervated by neurons expressing a receptor that is so far unknown. DA2 fits the bill. It could be what we're looking for. So OR56 has no known ligands, although people have tried a lot to find a ligand for this. Here is again the antenna in a close-up. So now we're going to start looking at all these sensilla, all these little hairs sitting on the antenna there. Single sensillum recordings, we plug in with an electrode at the base of sensilla. These sensilla are about 2-3 microns thick, about 50 microns long, and then we record from them. Here is this sensillum expressing the neuron, or housing the neuron expressing the OR56 receptor. You can hook it up with GFP, and then you see where they are on the antenna. And then you can record from them, and lo and behold, we get a very nice response to geosmin from the small spiking cell. Here is the statistics. So very, very strong response to geosmin. So our hypothesis then, of course, went, well, these neurons are probably the sole input, but we had to prove it, right? So the antennal lobe contains a lot of glomeruli. We maybe not see them all from the calcium imaging, so we have to go on looking at these other ones that we might not have seen. So we need to check the rest. A lot of more single sensillum recordings, checking 39 different neuron types, expressing 43 different receptors in all of these different sensilla, a lot of recordings, a lot of electrophysiology. And then they were screened with 50 diagnostic odors, and what do we find? One single neuron responding to geosmin. There is absolutely no response in any of the other neurons on the whole antenna. We have checked them all. There are 1,200. Okay, so maybe the sole input, right? But another control, of course, is a behavioral one. If this sensillum is really the sole input, if we shut it down, then we should also abolish the repulsion to this compound. So we went to the next level using Shibire. Shibire is a temperature-sensitive mutant dynamine. That means that you shut down synapses. So when it's 25 degrees, the synapses work very well. If it's 32, they're shut down. So we went to our system, we expressed Shibire in our flies, we did 25 degrees, 32 degrees, we did our behavior, and when we did this, we shut down attraction totally. So yellow is at 25 degrees. When we increase to 32, we abolish the effect of geosmin. So geosmin seems to be totally dependent on this. The controls, they are still working. So again, a proof that this is the sole channel. Great. Another control that actually came to me last week while I was in Chile, we expressed a receptor for a highly attractive odor in the geosmin neuron. So now we can excite both attractive neuron and repulsive neuron at the same time. So we should shut down, right? And that's exactly what we see. So when we stimulate normal flies, we get very strong attraction to this odor, but when we express the receptor for this odor in the stop channel as well, then we get full stop. So another control that this is really totally dependent on geosmin. So how specific and sensitive are then these OSN? Back to single sensillum recordings, 120 compounds, testing them to see, is this neuron really specific to geosmin, or will it respond to a lot of different things? And it's amazingly specific. You see here, these are all these 120 odorants, and basically the take-home message is that this neuron only responds to geosmin, nothing else. Totally specific to one single compound. When we look at the sensitivity, it starts at 10 to the minus 8. That is homeopathic concentrations, very, very low concentration for olfaction. So indeed, it's a remarkable selectivity, remarkable sensitivity. It actually only compares to what has been seen in moths when it comes to sex pheromones before. Really amazing system for detecting odors. What about the next level then? If we look into the brain, into the antennal lobe, of course this message has to be transferred to the next level. Blue is coming in, green is going out. So now we're looking at the green neurons. We went in, we did a bunch of in situ in vivo patch clamp on these PNs, hundreds of them to find the right one. And after about 2 years of work, finally we found our neuron. This is the neuron. And if you look at all these recordings here, you see that it's only responding to geosmin, nothing else. Again, something in parallel at the next level in the brain. And then if we compare this to morphology, we found that it is the DA2. It's the same glomerulus. So there is something coming in, there is something picking it up, and these two have exactly the same specificity. So it's a label line going through the brain, a label line for geosmin, which is detected with this amazing specificity and sensitivity. So why? We're still asking, why do they need to detect this with such a specificity? So we started thinking, why? And then we looked at these organisms that we had before that emit geosmin. It's these fungi here. Why are they, well, not why, but why do the flies respond so strongly to this compound? So as neuroethologists, of course, we had to go back and look in the lab and say, do flies survive on these microbes? Because the normal thing that flies live on is yeast. We think that they eat fruit. They're called fruit flies, but the real correct name is vinegar flies because they live on yeast. They eat microbes, they eat yeast. So we did the comparison. We fed them yeast and we fed them streptomyces. And when we look at this in the lab, they die. They die from the streptomyces. It's bad things for them. So if you look at this, which doesn't look so nice to us, it's also bad juju for the flies. They will die if they oviposit or if they eat from this orange. So basically, if a female is smart evolutionarily, she should not oviposit on this orange, right? So we needed to test that. So we went to the lab again, we put this fungus in a Petri dish, and we let the flies oviposit. And we could see that they really hate ovipositing on things that have the smell of streptomyces on it. Okay, how do we control for this? Well, then we went to our microbial colleagues again, and they gave us a very nice mutant of this fungus, which does everything normal, but it doesn't produce geosmin. It's the only thing. An enzyme is shut down that produces geosmin. Okay, do the flies do their thing then? No, they oviposit normally. And the larvae die. So it's very stupid to do it. But here you don't have the key compound. And in this way we can show that it is geosmin that does this. It's the only thing that is responsible for this behavior. We could of course test it also by trying the compound itself, and that also shuts down oviposition. So we have sort of a triple control that this is really true. If we try benzaldehyde, the old super negative compound, it has no effect at all. So geosmin is something really special to these guys. So in short, the smell of this fungus, it signs something that is really bad for the flies, some bad food. It identifies inferior or even toxic food sources to the flies. And this kind of identification is totally dependent on geosmin, the presence of geosmin, and geosmin